for accommodating your schedule. I know that you don't usually meet on Mondays, so I really appreciate that you're here. And it's really a pleasure. I've never been to UCLA. I went to school at UC Davis. I did a postdoc at San Diego. I did almost a leapfrog LA all those years. But really, I mean, I think except for UC Merced, I've been to every single UC campus, not UCLA. So I'm looking forward to seeing you in the afternoon at this wonderful campus. I know that you're having a great day. And thank you for being here to listen to our little story on the almanacs of Argentina. And like the title says, I mean, it encompasses a lot. The talk is really going to be a little bit of a summary of what we've learned about monogamy in almanacs by looking at some of the factors that at least historically have been proposed as possibly influencing evolution, the maintenance, the functioning of monogamy in all kinds of animals, not just in primates. How did I get to this? So I studied monogamy since I studied grad school at UC Davis, working with TV monkeys, with Sally Mendoza and Bill Mason. And I really look at my interest is in focusing on mating and social systems. But what I've done all these years is always looking at taxa where you just have one adult male and one adult female reproducing with each other. So for the purposes of our conversation here, that's going to be my kind of definition of what I mean by social monogamy. And in trying to understand what you find in a primate, a mammal, a male, limiting themselves to reproducing with one female, I draw from different theories. I was trained in animal behavior, but working with psychologists. So there is a little bit of my thinking in looking at socio-ecological theory, and that's going to be an important part of the talk, try to understand what are some of the ecological factors that may make monogamy and pair bonds work, right? We go out in the field and we notice something about the ecology, the forest, and we notice that these primates are only in monogamous groups. What are some of those ecological factors? We'll come back to that. When we see that, when we see just one male, one female, we notice that sometimes they're different, sometimes they're not, right? And so we start going from sexual selection theory and hoping to make some interesting contributions to paleoanthropology. We're looking at living primates, we're examining sexual dimorphism, trying to relate that to differences in morphology and behavior, hoping to contribute to understanding what we were like a couple of million years ago. We look at those differences and we notice differences in morphology, we notice differences in behavior, we notice difference in behavior and in patterns of care, for example. So we become very interested in fatherhood. That's myself with my 3 boys doing a very much male activity of watching a soccer game. So we draw from parental investment theory to try to make sense. Why is it that not only you have monogamous primates, but now you have something like our monkey or TV monkey, where you have so much commitment by the male to providing care to the infants. So through all this, we're really addressing some important questions in evolution anthropology. Pair bonds. I mean, there's some consensus that at some point in our history, they may have been important. Right? What are the reasons why we end up in this pair-bonded relationship? Love, as we call it in the magazine, as it is in the title. This very, very emotional, strong connection between one woman and one man. What are the reasons for that? How do we relate sexual dimorphism in anatomy, morphology to behavior? Another important question. And then also patterns of care, another possible very relevant factor in the evolution of human societies. It's been an exciting couple of years. If you're doing monogamy, it's been an exciting couple of years. There have been a couple of papers published in high-impact journals discussing what were the factors driving the evolution of social monogamy. Lucas and Clattenburg published an important review of 2,500 mammal species looking at the evolution of social monogamy. There was another paper that came out on the same day by Opie and colleagues looking at social monogamy in primates. That group was just not looking at social monogamy in primates, but claiming a link to male infanticide. And that was followed by a long debate in some high-impact journals about who is right, who is wrong. What I want to do today is contribute our little something, actually working with monogamous primates in the field, to evaluate some of the ideas, hypotheses, and data that those papers have brought up to light. And I think there is a need to do that. There's a need for us to continue collecting solid data on a few of those species that are then brought up into these wide reviews. We can discuss that later on. I have my significant reservations about these wide taxonomic reviews. This is just to illustrate why I think that studies of individual species are crucial for this dialogue. One of the paper claims that owl monkeys typically give birth to twins, and they cite Clayman 77. Poor Deborah Clayman, she was really saying that a Yodos only bears single young at a time. So you really need to be careful with these wide taxonomic reviews of so many species at one time. What we've done is taken a few taxa, all of them socially monogamous. And over the years, a little bit in Argentina, a little bit in Ecuador with my colleague and friend Tony Di Fiore, who is now in anthropology at Austin. We have 4 taxa. All of them are found in socially monogamous groups. We're studying Saki monkeys in Tipitini, Yasuni National Park. We're studying titty monkeys as well at Yasuni. We studied Eotus vociferans for a while at Tipitini. We're not really doing any work with them now. And then I've been studying Eotus azarai in Argentina for a longer time, and that's going to be my focus for the remaining of the talk. The Almaquí project of Argentina that we started back in 1996. If you want to learn more about the project, we have lots of opportunities. We were discussing with Susan how much help we need. So there are always opportunities for collaborators, for students, grad students, volunteers, undergrads. So I invite you, the young people in the audience. My students told me that I had to have a Facebook page, so you can browse it. You can look at our website as well and learn a little bit more about the owl monkeys. Because I want to try to provide you a review of much of what we've done in the last 2 years. I'm not going to go in a lot of detail into many topics. But we will have time later, so we can go back into some more detail. I think there's going to be a little bit of everything for everyone in the room. Owl monkeys from Panama down to Argentina. And contrary to what some recent reviews may lead you to believe, no, there isn't just one species. We recognize 11 species. We're focusing on Eotus azarai, found in Bolivia, Paraguay, and Argentina. My work is going to take place at the southern end of their distribution, northern Argentina. So if you're a student, you're looking at the evolution of primate societies, please disregard everything they say about Eotus trivirgatus in the review, in the lab table. They say that it's only been revised recently. They were revised 30 years ago by Hershkovitz. At that time, 30 years ago, we knew we had 11 species. So it hasn't gone through the rapid speciation that some other taxa have gone overnight. But anything you look at Eotus trivirgatus, you really want to be careful, because at that time we were lumping information from all kinds of species into just trivirgatus. We recognize now 11 species. We've recognized, for 30 years, 11 species. After I worked with titty monkeys at Davis, I went to Argentina, and I wanted to continue working on monogamy. So I found a private ranch in the province of Formosa, the White Collect Ranch, and there we established camp. It's a subtropical forest. We're barely outside the tropics, 25 degrees south. It's a relatively homogeneous forest, but only grows along rivers. Now, this is not a fragmented forest. This is not like Lomas Barbudal. It's always been like this. Since the forests grew there, maybe 10,000 years ago only, it's always been like this, where you have big grasslands in between these gallery forests along the rivers. So dark green is where you're going to find owl monkeys, including these patches. And all these are natural patches. A wonderful, wonderful system to look at private species functioning in a mosaic, in a fragmented landscape, not created by human action. At that site, we established camp that over the years has been getting a little bit better. I wouldn't say that there's a lot of comfort, but we're doing a little bit better than before, so we can offer you some facilities for being protected from the weather, but we still sleep in tents. But now we have a house, half hour from camp, half hour from the ranch, Casa 100, and there we have some wonderful library and an area for people to rest, to do data management. We have our labs, we have our computers. More and more, I'm sure that Susan will agree with this, more and more it's becoming an issue of how you manage your data. So those days in which you would only spend time in the forest, at least for us, they are gone. If you don't go back and do a lot of work on your data pretty much right after, you have a mess very soon. And we have, I have an amazing team. We have 4 people, we have 4 permanent staff members down there every day. And mainly Marcelo, Cecilia, and Victor, who are from Formosa, and been working with the project for 10, 15 years. And then huge number of assistants and volunteers who've been instrumental in everything I'm going to show you. Now, they're right there now while I'm here talking. And lots of collaborators that I'm going to be mentioning as we go along. And they've allowed us to do this. So very much like you know so well from the Capuchin project, we've been sampling year-round. So we're now looking at the 3rd generation of identified individuals. And this is to give you a sense of what we can do down there. We keep an eye on 25, 30 social groups. We keep an eye, right? We're not doing intensive monitoring. But I want to say that between 15, 20 of those we monitor at least monthly. Our behavior is focused more like on 5 or 10 groups that we can really keep habituated. And we have life history data from roughly 200 animals that we have captured. When you put that in numbers, to give you a sense of the intensity of this, it means that for 18 years, we've had contact with the monkeys 4 hours a day, every single day, 7 days a week. You always have someone up there 4 hours. What have we learned? Or what did you know before our monkeys came to the scene? If you knew something about our monkeys, it was probably this, right? They're nocturnal. I have very little chances of actually studying them well. And it is true. I mean, our monkeys are strictly nocturnal, all the way from Panama down to northern Bolivia. When we studied them in Ecuador, when Patricia Wright studied them in Peru, they go to sleep early at dawn. They come out of their tree holes early at dusk. You have very little chances of seeing them during the day. But in Paraguay and Argentina, our monkeys are cathemeral. Cathemeral, a word coined by Ian Tordersall in the late 80s. What I'm saying here is that this is a species that will be active both during the day and the night. And I'll show you a slide next that will get into some more detail. But you can come and see our monkeys like that, basking in the sun. In this case, they are eating flowers from a trumpeter tree that I know you have here in California. It's a fascinating activity pattern. I'm going to walk you slowly through this, unless we have a whole bunch of chronobiologists in the audience. I would assume we don't. So this is a very, very rich slide with lots of information. This is what the circadian biologists, the chronobiologists call a dual plot actogram. And so we have on the left for the summer, on the right for the winter. And it plots 48 hours of data from midnight to midnight, and again, the same data. It's for visual effects, you plot it twice. Now, what I'm showing you here is data for just one owl monkey who was fitted with a collar that records movement continuously, 24/7, for 6-8 months. But here I'm just showing you the records from day 1 to day 90. 3 months of recording for one owl monkey ranging freely in the forest. And what it shows you right away in the summer is that the owl monkey has 2 peaks of activity. I mean, black means the animal is moving, right? On day 1, there's more activity there. Activity slows down, yeah? So there's more activity right after dusk. There's another peak of activity early at dawn, a bimodal pattern of activity. You compare this with this, and you see how activity is slower during the winter than the summer, and how there's more activity during the day hours in the winter than the summer. Yeah, are you with me? And then what's fascinating that this little device will show you are the effects of moonlight. Can you see those? Effects of moonlight are reflected in these diagonal bands. So when there's a new moon on the right, I'm showing you the new moon, there's very little activity during the night. When there's a full moon, there's a lot of activity. But activity is not constant because the animals are really active when the moon is out. The first, if you're out there, if you like the moon, if you're a night person, the first few nights of a full moon, the moon is out the first hours of the night. And then it sets. Towards the end of your full moon period, the moon comes up late at night, and so you have more black towards the second half of the night. That's why you get the diagonal band. So strong effects of moonlight, strong effects of temperature, and light. The animals have a bimodal peak of activity, peaks at dawn and dusk, more activity when it's warm than cold, more activity when there's a moon than there isn't. Another important point about almanac biology, there's very little sexual dimorphism. And this is, I mean, I swear, this is no exaggeration. We have mis-sexed animals laying on the table. I mean, anesthetized, we examine their genitalia, we score them as, I always get these two confused, we score them as males, and then somebody can later cross it and score them as females. We had to hear back from the San Diego Zoo with the genetics to confirm that John was really Mary or vice versa. Actually, no, I do remember. The problem what happens is that their penis and clitoris and their testes and labia are so similar that under the effects of ketamine, of the anesthetic, the testes retract, lower temperature, and so animals that we had sex as males, then quickly, tired at night, mosquitoes, you want to go home, somebody reassessed and called female. But very, very little dimorphism at all. I mean, this is just, I think this is done. A very little dimorphism in body mass, in size, in canines, in everything. Their territory, yes?
Are there behavioral differences when you watch them?
There are behavioral differences. If you can hold on to that question, it's going to be part of the talk later on. There are some behavioral differences, but nothing very conspicuous. There's some division of labor. We'll get to that later on. They're territorial, so each group, there I'm showing you 18 groups, the ranges of those groups. Each group has a core area that they pretty much use exclusively, and part of that area that overlaps with neighbors. We'll talk more about that later. And they have intense paternal care. If you're not convinced by the cute picture of the dad transporting the male, I'll show you data. So these are data showing the proportion of times, of sampling points, but let's call it time, how frequently the male is seen transporting the infant. And what you can see is that except for the first week on the x-axis, when the male does very little, after that, infants are pretty much on the back of dad the whole time. Yeah, it goes down, by week 15, by 4 months of age, now the infants are pretty much independent, and they're not neither on mom or dad. But heavy participation of the male. 90% of the time, the infants are being transported by dad, not by mom. And they're monogamous, socially monogamous. Every single report of any owl monkey species from Panama down to Argentina, studied for a week, studied for years, again and again, you just find one adult male, one adult female, and some young. I'm not calling them their offspring, some young, but never more than one reproducing individual of each sex. Why? Right? So that's the question that we want to tackle today. Why are some primate monogamous? This is a wonderful graph that Peter Kapler published. It's a 2-page article, I think it's in Science, following those papers that came out. So we've known that for mammals, we've looked at how food, distribution of food, predation, disease may influence whether females are ranging solitarily or in groups. I mean, there's some consensus that ancestral mammals probably were solitary. Now the question is then what happened over time? What happened with male strategies to find those females? And there, the picture is not so clear. People have looked at the potential for infanticide as something that influences how males distribute themselves on distribution of females. Some people have looked at the potential for providing care, and other number of strategies by males, competition between males, coalitions by males. All of those should influence whether males are pairing with females, and then you get a monogamous group, or some other possibility where you don't get monogamy. But so it is possible, if we're trying to understand monogamy in primates, one way of doing it is beginning to understand something about the distribution of food and the use of space by males and females. And I want to share with you some data to evaluate that. So in a nutshell, this is your kind of textbook hypothesis for explaining monogamy, and it's not the kind of hypothesis that you can really evaluate to any satisfaction, but I look at it and I tell students this, I look at it as a way of organizing our thoughts and organizing the way we collect data. So maybe the spatial and temporal distribution of resources and females makes it difficult for males to monopolize more than one female. When that happens, when there's something about how food is distributed in space and time, yeah, maybe that poses a challenge for males to monitor more than one female. We can come up with some predictions that relate to what we should see if the spatial distribution of food is important in what males are doing and what females are doing as well. So one thing is that we wanted to look at is that what is the size of the ranges of owl monkeys? If we are in a forest that we know is relatively homogeneous, all things considered, I mean, it's not like you were saying that you have capuchins using forests that are very, very different. No, I mean, ours is a relatively homogeneous forest. Then, if food is important and the forest is homogeneous, we are expecting that the sizes of those ranges will be more or less similar, right? We would not expect to see ranges that are 100 times larger for one group than for other. When we look at that, collecting data over the years, so what we have is ranging data. Each time we go and we find a group, we record where they are, and so over the years, you have data on some 20, 25 groups, from 97 to 14, the number of times you've seen them, you plot those data for the groups that you feel more comfortable with those data, and you produce some summary stats for the sizes of those ranges. Differences are significant, I think. The 90% home range is on average 6 hectares, but it ranges from 4 to 11. So you really have groups that are using 3 times the area of other groups. The core area, the area where they spend most of their time, and your living room, so to speak, not the whole house, including the yard and that shit that you never go, your living room, now it's only 2 hectares, but still between 1 and 2.6 hectare range. So I don't really think, from looking at this, that I can say that the size of owl monkey ranges are very homogeneous. Now, maybe the size doesn't matter after all. It's the quality. And what really matters is how much food you have in those ranges, yes? Because it could be that even what we perceive as a homogeneous forest, it isn't for the monkeys. So we decided to look into the quality of those ranges. And what we did for that, I'm sorry, no, I'm getting ahead of myself. So that's prediction 3. Prediction 2 is that Lucas and Clattenburg, when they looked at the 2,500 mammal species, they noticed that you tend to have less overlap between the ranges of females in monogamous species. The idea being that if the females are spreading out because of food, there may be some competition, and a way to minimize that competition is by spreading out yourself from another female. So they compared overlap of ranges in solitary and monogamous species. And we find in owl monkeys huge overlap for the home ranges. Overlap of the kind that they find for solitary species, not for monogamous ones. But the story changes if you pay attention to the living room or the kitchen, more closer to what we're talking here. When you talk about the overlap of kitchen, then it's only 11%. So maybe there I'm starting to think or trying to sell you the idea that maybe there's something special, something different with the core areas that we don't have with the whole annual ranges. Now, to what I was saying before, we still want to look at the food in those ranges, right? Not just their size. Because maybe you have a larger home range, a smaller one, but the amount of food produced is similar. So that's what matters in terms of what females need to reproduce. And what we decided to do is I don't know how big is the range for those capuchins. I know that in Iguazu there are like 300 hectares. How big are they?
Well, like you're saying, it's immensely variable and it's so fragmented. It's
What,
really hard
but your smallest range?
I don't know. It's probably, it might be more like 200 or 300. I'm not sure.
The smallest. Right.
Yeah.
We have, so we can do this. You couldn't do this. We can take 4 ranges of 4 groups and map them entirely. We took 16 hectares, less than 10% of one of your ranges. We can do that. It's a simplified system, and easier for us to work in. And so we decided, let's take 4 ranges, make a grid, and map, tag, identify, measure every single tree in those 16 hectares. And so we did. We ended up with roughly 8,000 trees that we identified to species, so that we can really start getting a measure of potential food. I'm not talking real food. This is you going to the supermarket and recording everything that's out there. It doesn't mean you can afford it. It doesn't mean you're going to choose it. This is potential food, but it tells us something about the quality of the forest in each of those ranges. And what we found when we look at the annual range is that some of the larger, in red, the 2 largest ranges, you can see that they have more species of trees, many more individuals. This is abundance, 2,700 trees versus 1,100. And of course, this is expected. A total basal area that's more than double. Density is about the same. There are differences in the forest composition and structure between the large range and the small one. Same for some of those same differences apply to the core areas. Is the answer in the temporal distribution of food? We know the area is seasonal, right? And in the x-axis, we have the months of the year. In the y-axis, we have 2 measures. One for fruits. We go out there for 10 years every month. We've been going out there, and for roughly 400 trees, we count how many fruits there are in each of the crowns. So you count, you don't count the whole crown, you count a section, and then you extrapolate. And so you produce some estimates. You can see how the number of fruits varies over time, but there's a peak in our spring, September, October, November, going into the summer. And there's also a peak for, what are those, leaves, new leaves, and flowers. Everything blooms in the spring, in this spring of Argentina. Our monkeys are primarily fruit givers. 60–70% of their diet are fruits. So, one possibility is that if there's a time of scarcity, which we think there is because of these data, which is the winter, the dry season in Argentina, the months of May, June, July, maybe what we really need to be paying attention to is what are the resources that these monkeys have in their ranges during the dry season, because that's going to be the tough part of the year. You really need to secure enough resources then, right? Maybe the prediction is that if the dry season is a limiting factor, we need to see what kind of foods these ranges hold during that time. And we were very lucky. We decided to do this in 2008, and in 2008, we decided to do it in 2008, and we decided just to look at their foods in the winter. Because for our monkeys in a subtropical forest, if I can monitor every single individual of these 4 species, I'm getting a very, very complete view of their diet. Ficus, Chrysophyllum, Gonocarpum, Enterolobium, Wasuma. I don't know how many we have in the audience who look at trees more than monkeys. What we found is that the number of fruits varied, yes? Large territories, you can see, had many more fruits than D500. This is a territory that is only 4 hectares. So now we're not talking anymore potential food, we're talking real food. The food basket of D500, it's significantly smaller than the other groups. Same with the core areas. There's less food in the smaller territories. Now we're getting closer to identifying either a problem or a puzzle. How are they doing it if they have less food? On the following year, we found these data. which was very, very different. And what happened? We were not planning on this. These are the kind of things that nobody tells you, but really this is how science works. We didn't feel comfortable with the data we had for 2008. This was an NSF grant. We said, we really need to do it again. Let's replicate it. And we were so lucky that there was a drought between the two sampling periods. So the winter of 2009 came after a huge drought. And there were no fruits. You can see, compare the fruit production of 08 and 09. Fruit went down. Now, it went down, but it went down significantly when you look at the 80% range. The reduction in fruit abundance within the core areas isn't as pronounced. And the differences between the ranges on a harsh year, at a harsh time of the year, they're reduced. So we're beginning to imagine that you're getting here at the point where you're looking at really what's the minimum requirements for all monkeys. Harsh part of the year on a harsh year. You're still getting what you need from your core areas. And how do you do that? Well, among other things, you do it by adjusting your diet. On the top panel, you have the diet from 2008. You can see data supporting what I said before. All monkeys are primarily frugivorous. Yes? But what happens when there are no fruits? Well, no surprise. I mean, animals are not optimal foragers, or they are optimal foragers in the sense that they will eat whatever they can. So they switched. And all groups included many more leaves in their diets than they had before. Any questions up to here? I mean, we're a small crowd. If anybody has, I don't want to lose anyone in the process. So if you have any questions, please feel free to bring them up. Now, we've talked about the use of space. We've talked about potential food and real food. But if we want to build an argument around why all monkeys are monogamous, we really need to get to how this group does in terms of success compared to that group. Why are females spread out? Why is this male there and not here? So we really need some measure of reproductive success, because food and space are all only proxies for trying to guess what their success will be. If the resources are determining the distribution of females, and therefore males, we can predict that females will have higher success where resources are more abundant. So it's the variation in resource availability that should predict variation in success. So our question was, does any of this variation in the size of the ranges, in the amount of potential food, does it match any variation in the success of the groups? And for that we looked at our life history data. So like I said before, we have a number of groups that I call here core that we monitor more intensively. And so you can see that we have many birth seasons, and this is the number of babies born, and the number of babies that disappeared, and the age when the young dispersed. And then we have another set of groups on which I may not be as willing to bet my money. I mean, the data, they are more equivocal, or the ranges of error are bigger. But we have all these data to look at things like infant production as a proxy of success. Yeah, you can think of, really, we're trying to get something about the ranges. I mean, is this range more productive than that range? Are infants dying more there? Are juveniles dispersing earlier because there's no food, or later because there's still food at home? Well, here I'm showing you just the data for infant production. The number of years, the percentage of years in which the group in that range had a baby, related in relationship to the size of the core area. And you can see on purpose, I try not to draw lines in those graphs. I mean, I could have drawn a line, but I don't see any strong relationship there. No matter what the size of the core area is, there is fluctuation in how likely we were to find a baby in those ranges. Are you with me? That's infant production. I'm not going to graph all the other data. Let me just show you this table. Whether you look at infant production or infant mortality or age at dispersal, any of those proxies, we just don't find any strong relationships to the size of the range and or the food available. The strongest relationship is there, 0.4. And still, I mean, it fails to reach statistical significance, but that's a sample issue. but this is as strong as it gets, and there seems to be a relationship between age at dispersal and the size of the core area. Now the truth is you can make two very different hypotheses about that. I mean, if the core area is good, then if the core area increases, then I disperse older, yeah, because there's enough food for everyone. But if the core area increases in size, I may also be dispersing younger. So we know that there may be a relationship. It's so much harder to pinpoint on what may be the mechanism driving that relationship. So what can we say about how ranging patterns and resource distribution explain about social monogamy in our monkeys? So some take-home messages for this part of the talk are that there are marked differences in the sizes of the annual ranges and in the potential amount of food in those. So the resource base is not homogeneous, is not constant. Now, the overlap is less in the core area than the home ranges. And the differences in food availability in the core areas are smaller than the home ranges. So maybe in the future, as we try to explore this more, we need to start paying more attention to the core areas as the functional unit on which social monogamy in our monkeys may be functioning. Reproductive success, we're not finding anything, we're not finding the silver bullet. Am I convinced that there's no relationship between food and reproductive success? No. I'm sure there has to be some connection. But we need to keep looking because we're not finding anything that gives us the right answer. So, for the second part of the talk, I want to take the second part of the hypothesis. Monogamy may be explained by something about the distribution of resources, but only as long as it interferes with the ability of males to monopolize females, which is the second part of this. And we want to know, what are males doing? What can males do? For example, why don't we see, if I'm telling you that one range is 4 hectares and another one 12, why don't we see, I mean, let's take D500 there. It's a 4 hectare range. Why doesn't the male from D500 suddenly range over also E500, which would make it only a range as big as the range on this territory? Why don't we see males becoming polygynous? That's the other part of the hypothesis that we need to evaluate. We think that monogamy is maintained through mate guarding. We think that there are mechanisms of aggression that prevent males from approaching other males, that prevent females from approaching other females. We thought, I mean, actually now we know that there's intense competition between males, there's intense competition between females. And those are mechanisms that prevent that male from becoming polygynous. If he were to say, you know what, wait for me here, I'm going to go check on that other female, by the time he's gone, for one thing, the other male will not let him approach. And by the time he leaves, somebody else may come in. You really have to stick close to your partner. Why do you have to stick close to your partner in our monkeys, in our system? Because they are not just living in socially monogamous groups. That is part of the story. 25% of the story is that 25% of adults are not in those cute little groups, but they're living as floaters. These are your troublemakers. These are the young adults who disperse from groups and are floating around those territories trying to enter a group, challenging the resident adults, stealing reproductive positions in those groups. This happens. It happens for males, it happens for females, talking about differences in behavior. So, no differences in terms of feeding, no differences in terms of competition. Over 10 years, when we looked at records of 10 years, we found 27 times the resident adult female was replaced by a female floater. 23 times males were replaced. So there's a lot of competition, and that meant that the median duration of pairs was roughly 9 years. Yes?
Can the floater, male and female floater, establish new territory, or do you have to take over?
The answer is no, they cannot, because we have not seen them do it. But we have seen them I want to say maybe 10 times in all these years, we are walking around and we notice two animals. Remember, we cannot tell a male from a female. So we always make a joke that I'm assuming it's a male and a female, right? I mean, if we're ever going to find out homosexuality in animals, I don't know. But the truth is that we cannot tell. I mean, unless you're used to working with birds, or most people working with mammals and primates don't have that challenge. That you're looking at an animal, you don't know if it's a male or female. But we see these pairs, and sometimes they will come together. You see them for a day, for 2 days, hanging out together within the boundaries of another group. And then we stop seeing them. So we get the impression that they must be kicked out. We have never seen a group form de novo. The forest is saturated. So we only see animals moving into groups, kicking out the other ones. So that poses a challenge for you as a resident. That speaks of competition, that speaks of intrasexual selection, that speaks of you really having to guard your partner.
Can I just ask?
Yes.
If you can't tell the difference between males and females, how do you get these data?
Only through animals that we have captured, marked, and radio coded.
Right.
So when I said before that we have data on roughly 170 owl monkeys, is because for life history data we can only go with those 170 animals that we have captured. Yeah. The other ones, we're finally now learning how to sex them through poop samples, and that essay is running well. So we haven't figured out how we can sex animals. Actually, I have a grad student looking at this person, and she's been collecting all these data from these floaters, and she just got back to town, and but she didn't know how many males and females she had been collecting data from. So in a way, it's a wonderful blind experiment. You are collecting data non-biased. The problem is that you cannot produce a sample that you're sure it's going to be biased. So that's how we do it. Yeah. But we have lots of animals for which we have data that we cannot sex. There's a cost, big cost, of somebody forcing you out. That graph shows you the reproductive success of animals on the left who managed to stay with the same partner through their lives. And you can see that on average they produce 25% more offspring than animals that were forced to change partners. So there is serial monogamy, yes, but forced by others. We do not have divorce in the sense that people studying birds describe divorce. So because of this, one of the big questions of social monogamy is solved. I mean, males may not have a choice to leave. If you leave, you lose the opportunity to mate with a single female. That may be your best option, limiting your reproduction to just one female. But what it doesn't tell you is why males help. Because we have a lot of species, socially monogamous species. Still, many of those species, you don't see paternal care. So the question is, if the male is staying, because that increases, he's more successful with one partner than two over a lifetime. If he's staying, why should he provide paternal care? Is male guarding working is the answer. I mean, he should provide paternal care if you tell me that that is my son or daughter. If you guarantee me father certainty, I'll help. But if you don't tell me that, why should I invest? If mate guarding is working, if that mate guarding increases father certainty over a threshold, then we should predict to see paternal care. For that, we needed genetic samples. That's the kind of thing we do for getting sexes and biological samples. So we genotyped 128 individuals in 29 groups or floaters for 14 genetic loci. It's not a very diverse population, so you can see that there's an average of 4.3 alleles, but the combination of roughly 4 alleles per locus and the 14 genetic loci allowed us to discriminate paternity and maternity for 35 infants in 17 pairs. And the good news for that, or the exciting, I don't know if good or not good, I'm not going to have a moral judgment on the sexual faithfulness of owl monkeys. But so maybe the exciting news was that owl monkeys ended up being the first primate for which we have solid data showing genetic monogamy, not just social monogamy, but genetic monogamy. All 35 infants we could assign to the putative father in the group, and there was no other male in the candidate pool that could be the matching father. Of course, this can change tomorrow when we add infant number 36. And we're going through those data now. But it was very exciting because it allowed us to make a lot of sense of paternal care. This is really an extreme form of paternal care. And knowing that these are the biological offspring of the male, it helps understand why you have this extreme commitment. So, to wrap it up, we've made some progress. We're now looking at some other possible factors influencing social monogamy in monkeys. We're looking at the potential risk of infanticide. We have no evidence for it, but we're exploring our life history data. And we definitely want to do some more work on some of the strategies of the floaters. I think there's a lot to be learned from understanding the pressure that those floaters are putting on the population and the ratio of floaters to animals in groups. Floaters are very difficult to find, very difficult to even get numbers. I said 25% of the population, but that is an estimate that I would like to be able to reassess and improve on. But we have a lot of work to do, so we've done a little bit with food, but I think there's a lot of work to do with that. Ranging as well. I mean, anyone who's done ranging data with primates knows how difficult it is to get proper data. So I really want to close with a wonderful quote from a behavioral ecologist who studied fish. Some of you, I'm sure, you know him because you're in California. I'm just stealing his quote to conclude this talk. So it's been 30 years and we're still doing the same. I mean, it seems like no matter how much data we get, answers are never conclusive. So with that, I want to thank you. And I think we have time for questions, so I'd be delighted to take them.
Eduardo, are they just sitting in trees at night, or are they in holes? And is that a limited resource? And where do they mate? Do they mate at night in the dark in the holes, or out in the light?
In the places where they have been studied at night. So the few studies we've had of owl monkeys, none of them in more than a year. Patricia Wright in Peru, our study in Ecuador, some sites in Bolivia, all those species sleep inside holes. Now the tropical species, we published a paper showing that size has increased. Our guides are roughly 1,200 grams. The tropical species are more between 700 and 900. So for some species, you're talking 50% increase in size. The tropical ones are all inside holes during the day. Our species never ever, we've never seen them going into a hole. So they are not sleeping during the night, they are also active. They are active both day and night. So of course, a significant gap in our knowledge is getting more details of what happens at night. We did some studies at the beginning, but it's virtually impossible to collect data on behavior at night. You end up just with a number of saying moving, resting, foraging, and even that. So that's the answer for our guys sleep in open branches. They don't go into holes. Depending on the temperature, they may be huddling with each other, or they may be spread out if it's very warm. Where do they mate? We see them mating in open branches, no particular area. And what else did you ask?
Yeah, well, I mean, I guess that mating in multiple places means that a floater could possibly sneak in and get it.
Absolutely, and we've never seen those. Of course, I have no reason to believe that they would be more frequent at night than during the day, since we pretty much have seen the full behavioral repertoire during the day.
Well, you've seen the genetics too. So do they sleep in the
genetics,
same?
right?
Do they sleep in the same tree?
No. That's something else that we found that's different from what Pat reported, but different species, of course. They use many trees. Sometimes they will use the same tree for a few days, but they have many sleeping trees. Anyhow, for many, let me think. We're just finishing a paper looking at sleeping tree patterns and in 4 or 5 groups, and over the course of a year, we ended up with 130 sleeping trees for all 5 groups. So that makes it 20, 25 sleeping trees over the course of the year.
So that's not the limiting resource.
No.
that is
No,
driving
right.
monogamy.
Uh,
Okay.
But we never see, so the floaters, that, if you ask me, I think that we're going to find eventually extra pair copulations. What happens with those floaters is that they approach the group, and eventually, initially, there's a lot of aggression from everyone, not just from the same sex adult. The whole group reacts aggressively to the floater. Even some of the young, not very young, like your 2 or 3 year olds sub-adult reacts aggressively. But it's like they force themselves in eventually. I mean, they keep trying, and sometimes there's still fights, and then next thing you know, they're in the group, and the same-sex adult is out. Now, it's hard to believe that in that process, but we haven't seen attempts at copulating. Maybe there's so much running around and stress, and maybe even the mode is one of aggression and not love making. I don't know. We haven't seen it. Yes.
So the graph where you showed the loss in reproductive success for the males who had multiple partners instead of just the one. So that's just males that you're looking at there, right?
No, both.
It's both.
Both.
Okay.
Yes.
So females lose out through serial monogamy as well.
Yes. So it's very much like I use this when I teach my introductory lecture, and I talk about Monique's research with the pinwheel, and it's really serial. Who is doing better with this polyandry or polygyny? No, it's the same for both. It's the same for both.
And who's forced? So does one sex? Can you just explain a little bit more about the forced divorce and how it occurs?
If the floater, if the solitary is a female, it's going to be eventually the resident female that gets kicked out, and she may die. And the fate, if you're out, that's it for you. You may be alone for a few weeks, for a few months, but only once in all these years we've seen an evicted adult reintroducing himself, it was the male, into a group. So really, the picture that we're putting together is that it's a very slow, I'll get into some more detail, but let me give you a bit of a brush description of the life history. It's a slow development. So these guys are they are sub-adults, 2 or 3 years of age. They're not dispersing until they're 3.5 or 4, sometimes 5. You disperse, you're around trying to decide whether or not you're going to reproduce. We have no animals who have had a baby before they are 6 years of age. So now you're 6, you start producing babies. The animals who we know the longest have been around up until they are 14, 15 years. On average, animals are producing babies 3/4 of the time. So let's say you have the most 6, 7 kids, and after that, sooner or later you're evicted, and that's it. That's the picture that's emerging. Now, it's both males and females who are dispersing. Both sexes disperse, like in many other monogamous species. Both sexes are looking to enter a new group, and the way of doing it is kicking out the same-sex adult in that group. The cost comes mainly, this is something that's also been known for many monogamous species. Monogamous species take a long time to start reproducing. In birds, the idea is that there's some kind of courtship. Our monkeys consistently skip the first year. So this year, and really what you're losing there is that if I want to change, we don't know why or what's happening, but it's going to take us a year to pull our act together and start reproducing. And that accounts for pretty much that difference with the idea of staying with the same partner. They always skip the first season, and it has nothing to do with the timing of the replacement. Because the timing of the replacement, there's a peak. These replacements tend to occur in April, May, June, which is a very seasonal environment. Everyone's having a baby October, November, which means that everyone is conceiving May, June. Most replacements occur April, May. So there is time, but maybe there's no time to get to know each other. Maybe there's no time for something about the male and the female physiology connecting and start, because there's a lot of copulation. So it's not that they're not mating, they're not conceiving, or, well, I cannot assess conception, we will soon. They're not producing babies. I don't know if they're conceiving and the first conception aborts. We know that because we have the data.
So what happens to the juveniles when there's a replacement of a parent?
It depends. The replacement, I have a picture. I have a slide. Let me answer that with a slide that I have amongst one of the last ones. That one. The replacement brings lots of changes. This is just a sample of 6 groups where we saw replacements during those dates. And you can see how E500 had 6 animals. These are 6 groups that during the first few years we documented in great detail. So E500 had 6 animals, started to see fights. We didn't know that was the time. We're still beginning to identify them. But in that process, the female was evicted. We followed her for 2 months. She died. A 2-year-old, we found her right after the fights with serious wounds, bleeding, in very bad shape. She died. A second subadult disappeared. We never found her. And you see how it is not just a replacement. I mean, every group you lose 2 or 3 animals. Sometimes immediately through the fights, other times later on. Now, other times, we even published this, that one of the males, a new male came in and he ended up taking care of the infant who was already in the group. So I don't know what to make of that. We saw it once, but it happens. But this is a time of turmoil for the whole group. And many times, it's like the whole composition changes.
So for all the immigrating, maturing juveniles or becoming adults, they have to go through this process.
Yes,
of
yes.
Breaking up another
And the
unit.
subadult phase is really when we have the highest attrition, the highest mortality rates, which, you know how the field is, mortality, I mean, we don't know where they are. But that's where we really lose an awful lot of animals. It isn't within, our infant mortality / disappearance is in the order of 11%. But it's really when it comes, you're 3, 4 years old and you disperse, that we just lose track of the animals, and there has to be a lot of competition, a lot of challenges, and many animals don't make it. That's another, it would be great to find out ways in which we can better assess all the times that the intruders don't succeed. This is an extremely biased description of positive outcomes by intruders. All the other time, because we can notice the resident adult is not there. Sometimes we notice there's a fight and the resident adult is there, but so many times we don't notice the attempts. That's why I'm saying that we're still trying to come up with ways of better studying the flora population. Yes?
I remember reading that there are a lot of wounds that you can sort of see afterwards. Can you come into the group and see that there's wounding, but there's no replacement?
Yes, we see limping a lot. It is true. We looked at the wounds in the residents, and the animals expand, and we start seeing more and more wounds as the animals age, which we see as an indicator of these fights. And the wounds are, animals are missing canines, animals are missing whole chunks of earlobes. And I'm sorry, your second part of the question was, can you?
I think you pretty much answered everything.
So we see a lot of wounds, and we go there, and we recognize animals. Actually, that's one of the indicators that something happened, because we get to the group that we know well, and the adult male is limping, or the adult female is limping. And we know something. Before we didn't know. I mean, what's going on? Now we know that they fight, they fall to the ground, they break their bones, and many times they keep going. Yeah. Sure.
But even so, you can't say what distinguishes the successful takeovers from the unsuccessful attempts in terms of any of the characteristics of the pair.
No, I cannot yet. We do know we presented we gave a talk at AAPA last year. We looked at whatever we would look at, comparing the floaters and the adults. A very consistent difference is that the intruders are always younger than the residents, of course. So now we have a hard time assessing how that difference in age relates to some of the phenotypes that maybe will make you better competitor. Are they younger and stronger? Are they younger and healthier? And the only thing we know is that they're younger. Usually, because it's very hard for us, capturing these guys, I mean, it's a very complicated business. It's not like, so we don't have a mix of cross-sectional and longitudinal. We have a mix of cross-sectional, longitudinal, ad libitum, opportunistic, and whatever you can do. So I may capture a floater. I know its weight, or her weight, or her size, how good she is. Now she moves in 6 months later, a year later. I may or may not be able to capture her again. Or she moved in, but now she moved in into a group where I didn't know the male and the female. So it's a very messy data set that we are paying attention to, trying to improve. How do the new, how does the intruder and the resident compare?
It seems like one possibility is that they will acquiesce sometimes in the replacement of their partner by a newcomer when, I mean, sort of very speculatively, as a way of like increasing the genetic diversity of their offspring. I mean, maybe you've had enough kids with this partner, and now it's time to have the rest of your kids with some other.
Or the partner is getting old.
Yeah.
Or absolutely. Now the floaters, what I think is very clear is that the floaters are monitoring this big time. The reason we learn that is that when we capture them, and this is a little bit of an anecdote of how we work together with vets who have not been trained in these species, or they may not even have seen a forest. So the protocol said you capture your monkey, you bring it to the lab, you keep it there for as long as it's needed. If it's cold, you don't return it. If it has a little bit of a tweak in the nail, you wait until it heals. Right? So you pamper this animal and you only return it when you feel like this is it. And so we did that. Now, a few times, 2 or 3 at least, when we took back the animal, his place had been taken over. So they never made it back into the group. So you will return this wonderfully healthy animal who had now lost its social position in the group. So it's amazing how fast, because this was a matter of hours. The animals are moving in. So now we capture the animal, we do everything in the forest, we stay there, put with the group, we keep an eye, and the animals are up with the group. Yes?
Yeah, just sort of building on what Joe said, I'm wondering also if, because paternal care is so critical, whether the age of the infant has some effect on the likelihood that they'll acquiesce to an intruder. Because, so if they're going to be relative levels of codependence, depending on what the ratio of male to, not male to female care so much, but how important the paternal
if it
care is.
can have an effect on how likely the resident male is to defend?
Well, and maybe it'll only work if it's a new, so if paternal care is, so after week 1, let's say, so if at week 2 the paternal care is the highest and then it goes down, that as paternal care declines and the infant gets closer to independence, does that make the female more likely to, I don't know, I'm just wondering, well, I don't know how the conflict dynamic.
They don't overlap, because most conflicts with floaters tend to occur April, May, when most infants being born in October, November are going to be already October, November, December. So you already have a pretty much independent infant.
That's interesting, isn't it? That
Yeah, but first,
those things
we don't know what comes first. So we don't know if the intruders are trying later. We don't know. It's hard for us in the seasonal.
If they're trying at the other times and failing, or if they're...
Right.
Yeah.
For one thing, we don't know whether they're succeeding in April, May already, or they are trying in April, May
Yeah.
only. So that's, but we should have data on
Yeah,
that. Because even if we never looked at we have never quantified our data on how many times we've seen fights, even without knowing the outcome of whether there was a replacement or not. That may be a first way of getting a sense if the floaters are really trying through the year. But through the year, you're female. But that's the thing, I don't know if your willingness to engage in a fight and move in. Are you seeking a territory? Are you seeking a cycling female? Are you seeking a carrying male? We don't know. Sometimes we feel like you just need, I mean, it is tough out there. You just want to belong in a territory. But that's, I mean, I'm making that up. I don't know. I have no data to argue that is what's driving them. But we could look at, we have a lot to do with the floaters. It's such a big, it's exciting. I mean, for the first 4 years I published, there's a 2002 paper where I say that the probability of a floating population is unlikely, quote. So we've made some progress, but there's a lot to be done.
How often do you know where the floaters came from?
Now with the genetics, we're going to do that. We have this wonderful genetic data set, and it's going to be detective work. Sometimes we know because we put radio collars on the animals before they disperse, and they disperse, and you follow them, and bingo, you know exactly where they come from. Other times you know because we're learning, we learn quite a bit about dispersal patterns, and you don't know exactly where they come, but now we know that a floater moves back and forth for months between parents' land and some other area. So we can keep an eye on that.
So it's visiting before starting.
Yeah, the dispersal is not, it's not going to college one day.
Right.
There's many college trips
So
before.
I was under the impression from monogamous species that the emerging young would find another mate and just kind of find a territory of their own cut out of the forest. Are these unusual, or am I just wrong or about that being
Well,
the general pattern?
that was the teens on Tinasa 1981. We all grew up with one little study one year showing how this daughter inherited the territory from his parents. We've never seen that. We've never seen that with kitty monkeys in Tipitini either. But I can only say that's typical for these guys. I don't know what would happen in a forest where you would have areas where they can settle down. The times when we saw groups modifying their ranges significantly was not because a new pair settled in, but for some reason, and this happened only 2 or 3 times, territories are stable. Because another group, for some reason, extended its range. Why don't we ever see pairs trying to take over? Never. Why don't we see this pair that I was mentioning before say, no, you know what, we're staying. Instead of each of them going independently, why don't they, we don't see that. And I don't know. Unless maybe there's a lot of power to knowing the area and holding the trees or the sleeping sites or the escape routes.
Seems unlikely that we have small areas.
There's-
It wouldn't take that much to get to know you.
Yeah, but you're thinking as a human. Who knows? Maybe for these guys, 4 hectares, it's a lot. Yes.
I'm interested more about these replacements. So you see they sort of push themselves in, but is it a business between the two, same-sex breeding individuals, or do they fight with everyone in the group?
That's what I was saying, that sometimes we got the impression that everyone is engaged in pushing back to this intruder. But then eventually there are fights between the two same-sex individuals. Sometimes the process as witnessed by us may take 2 or 3 days. Other times a couple of hours. But I am sure this is just a part of the story. I am sure that the process of assessment is going on way early. Sometimes we notice that there are floaters around, and we see them, and we see one, we see other, but we don't see any fights. But they are there. That's something I always tell students. Remember that you're only seeing, smelling, and listening to this with human senses. And these guys are using our monkey senses. So we're only getting part of the story. And then we were fascinated by how fast these positions are taken. Once a male died, he was very old and sick, a couple of hours you have an animal moving in.
What's the typical age difference between the male and female? Is there any pattern for one to be older than the other?
We're trying to look at that, another mixed bag of data. No clear pattern has emerged yet. But I think that's part of the answer to Joe's question.
Right.
It'd be interesting if there's not a clear pattern, right? But I don't have solid data to answer that. Yes.
You said there's 11 species in this genus.
There were 11 species described by Hershkovitz in 1983. Since then, Susan Ford did another reevaluation primarily on cranial measurements and morphology. And then Anthony Rylance did another one in 2009. But for the most part, it stayed there.
Yeah. So is there comparative? I mean, that sounds like it's an opportunity for comparative, looking at differences across these different species, and different habitats.
You have to work with nocturnal species.
Oh, right.
We don't know. We studied them for 1 year at Tipitini, and we just could not do it. And
You didn't have any data?
We published one paper. We
Yeah.
put out one paper describing ranging patterns and some sleeping tree sites. And now they're slowly, they're young, very motivated people trying to get started with some behavioral studies of the Yoros lemurinos in Colombia. We have now one Colombian biologist training with us. I am talking to a PhD student who wants to start, but it's difficult because you don't even know what to advise them. It's like we are struggling to get funding, and now we're going to the more boring side of our job, but we're trying to get funding. I mean, I get stories about those are not, my grad student doing dispersal, she's going to have now data from 8 females and 6 males who dispersed, and she couldn't get an NSF because sample sizes are small. And so I feel, I never know what to say. I mean, you have these Colombian students who say, we want to start early merinos, and you just don't know what they can do that it allow them to move forward. Because you feel like, can this be studied?
Yeah, I mean, one thing I was wondering, probably nothing you can say about this, but you know, the socio-ecological perspective seems to take a purely sort of functional view, asking what what's the relationship between ecology and the behavior, and you know, if it's the case that these, you know, I guess I'm wondering how much evolutionary history might play a role here, where you know, they're in a sort of marginal environment. You said they've gotten larger. I mean, marginal might not be the right word, but if the majority of the adaptations happened in tropical environments, right? And then you see a sort of differently adapted species, I just wonder how much some of this might have to do with you start out being adapted to a particular environment in this rainforest type of environment, and then something about this new environment causes, you know, might
like for the floaters, or
well, just because I mean, some of the things that you're showing are just so, I mean, it seems strange. It seems
like what? Because the only things
well,
that
the pure monogamy, you know.
Oh, but that I'm saying that. Remember that it's 35 infants, and when you want to publish, you say this is the largest data set, and you're right. But you can also say it's only 35 infants. So. Yeah. I mean, it just. The floaters are popping up everywhere. We know there are floaters now with the Sakis in Ecuador. We have documented that there are floaters with the TV monkeys. One of the things is that we keep working on this. There have been no long-term studies. There have been no studies longer than a year of any of these taxa anywhere with identified sex individuals. So, is this because we have we're habitary, or is it because it's the first time that we know the sex and age of the individuals we're looking at?
I mean, I guess it's the kind of thing I'm thinking about is differences in the habitat, but also differences in the fact that they're now partially diurnal instead of nocturnal, because you can have a lot more sneaky stuff going on at night, and I wonder if they're doing more of this behavior during the day, if somehow it makes it harder for other individuals to sneak up on you, and that makes it easier to defend the territories, which then makes defense of the females easier, and it's harder to go off into somebody else's and have sneaky copulations and things like that. So if there might be some sort of effect of the fact that
that's an interesting
they
idea.
move towards more diurnality than
We are. Let me
typical
speak to your question in the only way that like I said, where we're trying to help with what we can with those trying to start research in other nocturnal species. What I am doing, and this started now 3-4 years ago, I am developing the program to include also research with both captive owl monkeys and titties. So in my mind, we will have owl monkeys, titties, and sakis in the wild, and owl monkeys and titties in captivity. Most of the research with captive animals will be with owl monkeys. We've done a tiny little bit with the titty monkeys, but it was primarily life history. But there are 2 facilities in the States. The Dumont Conservancy in Miami, they have outdoor enclosures, and they have 3 or 4 different owl monkeys. So it's regular temperature and light, but of course there are provision, etc. And then there's a facility in Texas where you have 4 species of owl monkeys in controlled lab conditions, and you have like 80 families total. So we are hoping that we will start some genetic work. Actually, I'm going from here to Houston to meet with Jeff Rogers. They're working on doing the full genome of owl monkeys. My idea is that can we find some phenotypes that will be amenable to be examined in captivity? We may not look at ranging, right? But there are things that we may be able to look at. Because if we start looking at phenotypes and we show huge changes in phenotypes that we can evaluate in captivity for SRI, that would be what you're saying.
Yeah.
Well, if all of this has changed, which you can measure in captivity, it may seem reasonable. There are things that you cannot measure and may have changed as well. So we want to look at things like color vision. We want to look at how their fur changes, both in structure, in thickness, in length. Does it change seasonally for these guys who are now adapted to subtropics or tropics? Size. So that's a whole other aspect of the program that I'm hoping we will be able to develop in the next many years. But if we start seeing all these changes, actually maybe you're even able to document what are the genetic changes we see in 10,000 years. And how could they be related to the things we're seeing in the field.
One more question. That you're talking about monogamy as a demographic thing, one male, one female. But what evidence do you have for emotional bonding? None.
In the field without monkeys, 0. And that's been a struggle for me in my writing, coming from Sally Mendoza's and Bill Mason's lab, because the way I was trained and the way that I always talk about pair bonding implied an emotional bond that I could assess not only with measure of affiliation, but with measures of distress. Now, as you well know, over the last 10, 15 years, the way that pair bonding and bond has been used in the field primatology literature has completely developed a new meaning. And so these days, a bond in the field primatology literature means two individuals are close to each other. So I mean, as good as ants are extremely bonded. I see them always side by side. So the truth is, we don't have data. That's another reason why we want to start doing experiments in captivity, to understand what are the behavioral mechanisms keeping these animals together or not, and understanding paternal care. I get very excited about the possibilities of really understanding what's going on with these males. What's going on, what's triggering the responses to infants. We never thought that these things would be going so well. We never thought we would be capturing 170 animals. And because we never thought that, we never evaluated behavioral responses of the animals to the capture of their partner and reunion. Because today I could be presenting to you 170 experiments of separation or reunion in the field. Now with a student who wants to do her PhD looking at pair bonding, I said, please, we have to start collecting data in a systematic way. we notice that the partner left behind gives a contact call, the famous hoot that our monkeys do. Continuously calls, and that's a call that we associate with trying to reestablish contact with your partner. But we know that there's this the similarities with city monkeys are fascinating, and these guys are together all the time. They're feeding together, they're traveling together. One moves, the other one moves. But emotions, that's, as humans we know, that's a whole different story. Do you have any ideas about what we could do?
When the floaters come in, how much does the opposite sex partner participate in the fight? And,
It's a mess.
or is he
When the
just,
floaters come in, you can barely keep up.
yeah.
It's branches moving, animals falling. It's very, very difficult. People who've not done this, why don't you do that? Well, it doesn't work like that. All of us know. Yeah, but I need tin can, I need Hollywood studio to come in. So the truth is that it's so difficult to get it. We tried to do playback experiments trying to trigger those kind of responses. And when we tried, it didn't work out well. It's a pending subject. I'm talking that all of you know. But I don't need to explain to you that these things sound better on paper than they are in the field when we're trying to collect the data.
Pairs that have been together for a while, do they ever seem like they're not getting along very well?
No. There's a lot of no. Not only that, we have not documented that. What's interesting is that only after a few weeks of the intruders coming in, we have a hard time telling a long-standing pair from a new one. So if I didn't tell you that that's a new pair, a few weeks into the relationship, they're just functioning like they have been together for years. At the level that we can describe in the field. So that's why, again, I want to have students studying behavior 2 meters from the animals and getting all these details, all these subtleties of interactions. We have one PhD student looking at olfactory communication and pair bonding in Texas. That's why I really feel I'm pushing for this back and forth between captive and wild animals to enrich both.
Sure.
Yes or no? Are newer, more newly formed pairs more susceptible to intruders?
Are newly formed pairs more susceptible to intruders?
Those forced divorce occur more commonly in pairs that haven't been together very long.
No, we don't have. The answer to that is we don't have many short tenures. Only once we saw in a period of 2 months, 3 females, but that's never again. In 2 months, we went through 3 females in one group, and the first 2 we found dead. So fights and then never again. So no, we don't have very short. Now, linking into climate change and who knows, we had a few years of huge number of replacements in 03 or 04 that worried us because we said, oh gee, are we messing up the system with our captures? It was after a few years of very intense capturing. And in 1999 we had a last El Nino event, and we had the highest infant production ever with 100% of the groups having babies. And so up until last year, I was waiting for the next opportunity to test my idea that 99 had produced lots of babies with these pairs. 3-4 years later, bringing the competition of floaters through the rules, and that was the replacements. We had El Nino last year, there were not a lot of babies, so there went my testing of the whole idea. But we have at least 2 periods when we get the impression that replacements went up, and we don't know what we can relate that to.
All right, on that note, we are out of time, but thank you for a great time.
Thank you for the opportunity to discuss this question. Thanks.